Yeah, today we're talking about BH3 profiling, which is a technique I've used a lot over the past year, which has been basically adapted into a flow cytometry, high throughput protocol, and we use it to assess how the apoptotic machinery in cells is operating and whether as a whole the pathway is functional or not. So just an overview of what I'll be talking about today. It's not going to be anything too in-depth in terms of the technique, just because the good thing about BH3 profiling is that essentially we're leveraging already standardized techniques, just leveraging it to use to analyze a specific pathway. But in brief, I'll be giving an overview of apoptosis, an introduction to the methodology, how the technique is run, and some experimental examples from previous papers and of my own work, and then talk a little bit of like how the BH3 framework could be potentially utilized by you guys in your research, and how the initial idea and concept can be kind of expanded outside of maybe just a flow cytometry protocol. So going into apoptosis, I'm sure you're all familiar. We're talking about programmed cell death, the means by which the body and cells become program themselves to essentially die. So I personally focus on lymphoma and non-Hodgkin's lymphoma and apoptosis in that disease and resistance therapies, but apoptotic dysfunction, so in essence, this technique could be important and useful to a lot of diseases and pathologies. Here we see just from a Nature review showing kind of the apoptotic sensitivity of different cells throughout the body in mouse and human, and basically you see when you're developing and in certain hematopoietic systems in the adult, you see a high apoptotic sensitivity because these cells are constantly being selected for, and aberrant cells or misdeveloped cells are being cleared from the body. But apoptosis is something that can affect, autoimmune disorders, as well as any sort of pathology involving reproductive organs, heart, lung, liver. So it's something that could be applicable to a lot of areas of research. When we talk about apoptosis, it's usually defined in two pathways. So this graphic here, we're looking at the extrinsic versus intrinsic pathway. The intrinsic pathway is kind of interchangeably used with the mitochondrial pathway because it occurs through the mitochondria. The BH3 profiling technique focuses mainly on the intrinsic pathway, but just to kind of overview the extrinsic and intrinsic pathway, the extrinsic pathway is generally considered the pathway by which external stimuli result in the induction of apoptosis. So a good example of that is the binding of, say, the FAST receptor with FAST ligand by a cytotoxic T cell to initiate the destruction of the cell. But in essence, and then the intrinsic pathway is activated by internal stimuli such as DNA damage, some sort of oxidative stress, ER stress, or metabolic changes. It's important to note that while we're analyzing the intrinsic pathway, they do eventually overlap when you get to the activation of BID and subsequent caspase activation, which recruits the apoptosome and then eventual cell destruction. So it's important to remember that extrinsic pathway could be important in your studies as well, but BH3 profile is mainly focusing on analyzing the functioning intrinsic pathway of a cell. So if we delve a little deeper in the intrinsic pathway, a lot of these proteins are going to be important to analyzing our samples that I'll describe in a little bit here. So in brief, we receive some sort of apoptotic stimuli, let's say DNA damage, And then we have some upstream signaling, notably by P53, which everyone knows. And then we see the activation of some BH3 proteins, we call them. So BH refers to BCL2 homology. So all of these proteins involved in this pathway are part of BCL2 family proteins, and BH means these proteins are sharing sequence regions that are similar between this group. They share between 1 and 4 regions. Now the BH3 only proteins, which are activators, are the most pro-apoptotic proteins, and BH3 region is the cell death region. So these BH3 only proteins are considered highly pro-apoptotic and the initiators of the intrinsic apoptotic pathway. So notably BIM, BID, and PUMA are activators, seen at number 2. So what happens after the signaling is the activators then lead to the activation of BAX and BAC, which co-localize and then form pores in the mitochondrial membrane. Now the pores formed by BAX-BAC lead to the mitochondrial outer membrane permeabilization and the release of cytochrome C from the membrane, mitochondrial membrane inside the mitochondria, and subsequent apoptosome formation and caspase activation. Now this process is inhibited by pro-survival proteins, BCL2, BCLW, BCLXL, and MCL1 most notably, which bind to our activators, sequester them, and don't allow them to initiate BAX-BAC activation and pore formation. There's another class of proteins here that's important, which is our sensitizers, which include BAD, NOXA, and HRK, as well as BH3 mimetics, which include small molecule inhibitors that specifically target pro-survival proteins. So these two groups aim to bind to the pro-survival proteins and thus not allow them to inhibit the activators from inducing MOMP. So essentially, what the BH3 profiling technique is, we're looking at these pro-apoptotic proteins, so our activators, sensitizers, and the pore-forming effectors, we call them BAX and BAC. We're looking at how they balance with our pro-survival proteins, because whichever is more favored in the cell is going to lead to either a survival phenotype or an apoptotic phenotype. And the way we measure this balance is by measuring the release of cytochrome C from the mitochondria of the cell. So as I'm going to mention, the important proteins for this assay that we're going to be looking at are our pro-apoptotic activators and effectors. And that's the BH3, as I mentioned before, is our activators. These are BH3 only proteins, and that's where the name BH3 profiling comes from, because we're looking at these highly pro-apoptotic proteins in the intrinsic pathway. So there's more proteins than this involved in the pathway. There's 15 plus BCL2 family members, but these are the most important ones for analyzing the apoptotic competency of our cells, including the anti-apoptotic BCL2, MCL1, BCLXL, and BCLW. And below here, I have a diagram basically showing some binding patterns of certain peptides and small molecule inhibitors, which I'll elaborate on a little bit, and how they bind with our anti-apoptotic proteins. So you can see that the anti-apoptotic proteins in most cases are really trying to bind BIM, BID, and PUMA, which are probably our 3 most important initiators of apoptosis in response to apoptotic stimuli. But this binding pattern is important in our assay, because essentially what we're going to use is, by looking at the different responses to specific peptides and inhibitors, we can kind of figure out where the cell is either inhibiting apoptosis or if there's any sort of other dysfunction. So when we talk about apoptosis and BH3 profiling, we like to use this term priming. So we're essentially saying, how primed is a cell? So what that means is, how close to apoptosis is this cell if we were to give it some sort of insult? And the metaphor that is used often in people using BH3 profiles, specifically from the Latai lab, who developed this technique, is an apoptotic cliff. So here I have a picture. This is Trolltunga, I believe. It's a famous cliff in Norway. Side note, this climber hanging off it here actually got in a lot of trouble for this photo for encouraging dangerous stunts, so he had to make a public apology for this, but it's a very cool photo. But the apoptotic cliff metaphor is essentially, if we imagine going over the cliff as apoptosis, how close a cell is to that cliff is its level of priming. So if a cell is, say, 5 steps away from that cliff, and we have some sort of insult like chemotherapy that maybe pushes it 10 steps forward, then that insult is going to lead to apoptosis in that cell. But if we have another group of cells that is, say, 15 steps back from the cliff, that insult is going to kill or push off the cliff the one that's in front of it, but the ones behind it are still going to survive. And basically, we want to know how close are our cells to the cliff, and is there anything we can do to get them closer to the cliff? So that's where BH3 profiling comes in. So this is a paper describing the current kind of flow cytometry technique that I'm using in the lab. So the BH3 profiling in general as a technique started actually as an ELISA technique, moved to flow cytometry, and it was developed by Jeremy Ryan and Anthony Latai in the Latai lab at Dana-Farber Cancer Institute, which is in Boston. So I defer to them in terms of anything related to BH3. They actually helped us start the protocol in our lab, but they have patented this, and so any more in-depth questions beyond my knowledge is deferred to them, as they are the experts in this. But so the main idea of BH3 profiling, as I said, is to expose cells to different peptides in the intrinsic apoptotic pathway, as well as small molecule inhibitors and other drugs. Look at the response of cytochrome C in response to specific parts of the pathway, and then use that to determine how cells are undergoing apoptosis, if they are at all, and how close they are to the apoptotic cliff I described. So we use cytochrome C because it's a good measure of MOMP, and it's been shown to be a reliable correlation to cell death and later apoptosis. There are some proteins that can inhibit cytochrome C's activation of the apoptosome, but mass release of cytochrome C is in cell lines is correlated well with cell viability studies. So we use it as it's a pretty reliable indicator of commitment to apoptosis. So the key is that we're insulting cells with our different peptides and analyzing how they are reacting to apoptosis without actually undergoing cell death completely, because exposing any sort of cell outside of culture to a drug or peptide or something, eventually it's going to initiate cell death over a certain amount of time. So it's important that we're looking at still live cells, how they are undergoing an apoptotic process in response. So the basic methodology for BH3 is you want to first create a profiling template with the it can be more specific to your lab, what you want to study, but basically you create a profiling template with the peptides that are the same sequences as the BH3 activator, effector, and sensitizer proteins, and also small molecule inhibitors to see how the anti-apoptotic proteins are involved in the process. Create first a template, which with you're going to expose your cells. Add cells and expose them individually in separate wells to peptides, and then measure the mitochondrial outer membrane permeabilization via cytochrome C release. I've included the link to the Latai lab. They actually have a whole section on their website basically detailing this entire protocol. They have an appendix with all the buffers and everything you need. So if you really are interested in trying a BH3 profile, I would recommend first going to their lab and just looking at the documentation they have. So the first thing to consider when if you want to do a BH3 profile is, do you want to do a plate versus a tube-based assay? I would advocate for a plate-based assay, especially because most, at the LDI we do, but in most places as well, they have a high throughput 96 well plate system that you can use with the cytometer. I would recommend that you go with the plate-based system because it allows you to use a larger panel and get more samples done in a more efficient time. Because as you'll see soon, when I describe my current panel, I have 26 different peptides that I need to expose cells to, so that's 26 wells. So I can do like 2 to 3 samples in one plate, and it's relatively quick and efficient, and I expose the cells for the same amount of time. Whereas if I had to do 70 tubes, it's a little more work, a little more time consuming. So I would advocate for the plate, but if you have an abbreviated panel, you could definitely use the tube. It's the same sort of principle. So when I'm describing the peptides or BH3 mimetics, these are the main peptides that you can use. So my lab currently uses BIM, BID, BAD, NOXA, and PUMA, as well as HRK and MS1. But basically, this is pulled straight from the documentation from the Latai lab, showing you that we're basically, they're truncated versions of the protein. So they're not full-length proteins, but they're replicating the BH3 or activating portion of these different proteins. And you can see these are the sequences. We use the same sequences, so if you want to use these, all you need to do is go to like BioBasic or some peptide company and submit this sequence and have them produce the peptide for you. It's relatively simple and straightforward. But some of these are like PUMA 2A we don't really use in our lab, but it's an inert version of PUMA. So there's different proteins you could use, but that's basically a negative control for, because you should see the opposite of your regular PUMA response. And then the small molecule inhibitors that we use to kind of look at the direct involvement of certain anti-apoptotic proteins include ABT199, also known as venetoclax, which is a BCL2 specific inhibitor, currently undergoing a lot of clinical trials in non-Hodgkin's lymphoma. It is approved for certain uses in CLL, chronic lymphocytic leukemia. There are BCL-XL specific inhibitors, WEHI539, as well as A1331582, which we use in the lab. And then ABT737, which inhibits BCL2, XL, as well as BCLW. This is a precursor to navitoclax, which went through clinical trials as well for use in non-Hodgkin's lymphoma, but eventually it stopped clinical trials because they found that it was it induced thrombocytopenia because platelets rely primarily on BCL-XL for survival. And then our important controls to assess whether or not the assay was done properly is DMSO, is our negative control for the mitochondrial membrane permeabilization and cytochrome C release, and alomethazine is our positive control, which induces the permeabilization of the membrane independent of the back-to-back effectors. So this is the current template I have for BH3. So basically, if you imagine these as wells in a plate, I divide my sample of cells between these different wells, which I have put varying concentrations of our BH3 activators, effectors, and sensitizers, and then the controls of alomethazine and DMSO. So these are the final concentrations, but it's nice to see a dose-dependent response to the activators, as well as we can use these higher dilution sample wells to kind of assess how primed the cell is, because if it's responding at low dilution or high dilutions, low concentrations, it means that cell is probably highly primed. And then if cells aren't responding to very, very high concentrations, we know that 100 micromolars is a huge concentration for BIM. So if a cell doesn't respond to that, like, we know that it is very, very pro-survival and resistant to apoptosis. So we use primarily BID and BIM for the activators, as well as PUMA, which is an activator, but also a pan sensitizer. It can activate backs and back, but it mainly serves to sequester anti-apoptotic proteins. And then as well as our other peptides, bad backs, or bad NOXA and HRK, and MS1 is MCL1 specific, BCL2, BCLXL. And then on the bottom, I have usually this third row we would use for any sort of combo therapies to see. One common one we do is MS1 with ABT199 to kind of explore the contribution of both MCL1 and BCL2 to our apoptotic thresholds. So this is just what my setup looks like. So basically, these are the peptides that I organize, and these are my stock concentrations. So then I dilute these into the final volume of my plate. But you would want to organize your stocks basically in a way that allows you to easily just go through each peptide, because you have to put the specific peptide or drug you're using in each well and just keep it organized. So to start, because I'm not going to go in depth with the flow cytometry part of BH3, namely because I think it's a technique that most people are familiar with, but essentially we're staining cells with fluorophores and activating those fluorophores and looking at the excitation of those through a flow cell. So you need to, one, identify your cells of interest and then measure cytochrome C. So for something like a homogeneous sample or a cell line, you really only need two colors. Your live dead, because we only want to assess apoptotic priming in live cells and not cells that have already undergone necrosis or already undergone apoptosis, and cytochrome C staining. However, for primary samples, obviously they're not homogeneous depending on what the sample you're looking at is. So you want to identify your cells of interest, and that'll be specific to your experiment or cells you're looking at. I personally use CD19, CD3, and CD5 a lot. CD19 being a B cell marker, CD3 being a T cell marker. So usually I'm looking at B cell populations. So I separate out the CD3, gate on CD19, and then sometimes use CD5 for certain diseases. For example, if I'm doing a CLL sample, CD5 positive cells are indicative of CLL. So I also use something like that. The cytochrome C antibody we use for the staining is listed here. Put the catalog and clone down. I've been using this antibody for a while now, and it stains very nicely. So I would recommend this antibody if you're going to try staining cytochrome C. So the cell staining is done, so the marker staining is done first, then we expose to the peptide, and then the cytochrome C staining is done after the exposure because it is an intracellular stain, so we need to fix the cells, and then we incubate with cytochrome C antibody. But the staining is pretty easy, relatively straightforward. Titrate your antibodies and just use the antibodies that will identify your personal cells of interest. Because you could assess the apoptotic priming. I usually do of B cells, but you could assess it of T cells or other cells in the body for sure. So peptide exposure, this is where I'm going to recommend for the plate assay as well as a multi-channel pipette. After you do your surface marker staining of your cells, then you're going to expose your peptides, expose your cells in equal amounts for each well to your different peptides or drugs in your protocol. So this time can vary between 30 and 90 minutes. So 30 minutes might be good for a primary sample that doesn't survive very long outside of culture. just because if you wait too long, then you might not see, there might just be too much cell death just because it's out of culture. Whereas 90 minutes can maybe better assess apoptotic priming in cells that are highly resistant to cell death. So I usually, if I'm working with cell lines, I do something like 90 minutes for our exposure to peptides. So you add your cells to your well, let it incubate. Most of the protocol is done at room temperature, except for maybe cell staining at 4 degrees for some of the cell markers you use. But I would recommend using a multi-channel because equal exposure to the peptides is important. So you want to make sure that you mix the wells very well, well, and that you are incubating your cells for the same amount of time. Because if you do it one by one, by the time you're done pipetting, the first well might be exposed for 120 minutes, whereas the other one might be exposed for only 30, and it's you're going to get varying results. The cell membranes must also be permealized using digitonin to allow the drugs to interact with the cell mitochondria. So usually when you're ready to expose your cells to peptide, we put them in a buffer with digitonin, mix them up, and then equally distribute them across the wells. And I usually prepare my peptides at 100x and then dilute down into each well to 1x. But there's many individual ways in terms of getting to your end concentration that your desired use is at. So then once we expose to the peptides, then it's time for intracellular staining of cytochrome C. So we recommend an overnight stain, but you could stain it for as long as an hour or two and analyze the results. But the Latai lab and we have found that the overnight staining does best in terms of getting a nice readout of cytochrome C. So in order to do the intracellular staining, we do, after the peptide exposure, we fix the cells with PFA. Then we stop the PFA reaction with a neutralizing buffer. Then we introduce our cytochrome C antibody with an intracellular staining buffer. So we're and then you the next day you come in. If you have an HTS like pictured here, you can put your plate on the HTS, have the cytometer read out your response of cytochrome C. If it's something like a cell line, you basically just need to gate on your live cells and then look at your cytochrome C response. Otherwise, you do an individual gating method for your cells of interest. If you have to use multiple, a lot of overlapping colors for identifying your cells of interest, you obviously want to prepare as well some compensation controls, which is kind of in that flow cytometry area that I'm not talking about too much. But so it's important to note that for cytochrome C, when we're looking at it on the cytometer, we're staining for cytochrome C retention because we're staining for what's in the cell. And the digitonin that was introduced in the peptide stage is the pores in the membrane are big enough that any cytochrome C released by peptide exposure is going to leave the cell completely. So a cell that is releasing cytochrome C will not stain positively at all. But we're usually concerned with apoptotic activation and cytochrome C release. So although we're staining for retention, when we portray our results, we usually do the inverse of the positive stain percentage and then convey it as a percentage of negative stain or release. So just to kind of summarize the typical BH3 workflow, I know it's kind of a lot. Hopefully you guys followed me here, but we basically start with a viability stain, then your cell marker staining, different for each experiment, peptide exposure, then our fixation, which stops the peptide reaction and prepares for our cytochrome C stain, neutralizing buffer, which stops the fixation, and then our overnight cytochrome C staining. So the whole, once you have everything organized, the whole thing takes about 3 to 4 hours, and then you analyze it the following day. You could analyze it same day if you're in a rush, but usually the cytochrome C stain works better overnight. You get a more sensitive result. So this is kind of, now I'm going to start detailing a little bit of the analysis and looking at some experimental examples. So this is a good example of gating strategy for a specific sample, where you essentially do gate your cells of interest based on cell size exclusion. So here I'm gating my lymphocytes. It's a pretty homogeneous sample, so it's pretty easy. And then we're gating our singlets out, and then I use aqua stain for my live dead. So anything staining positively is going to be dead cells, so I gate then on my live cells. And then here I use 786 as a CD3 marker and PerCP Cy5 as a CD19 marker. So here I'm gating CD3 positive or CD3 negative, CD19 positive B cells, which is pretty much the entire sample. And then you look at, so this is going to be the same for each well or each tube you're doing. Then the next step is to gate on your positive and negative controls and use those as your kind of gate guideline for assessing cytochrome C release percentages in all your experimental wells. So here we use DMSO, which can get kind of confusing talking about positive and negative controls, but DMSO is our positive control for cytochrome C retention, but our negative control for cytochrome C release. L-methazine, opposite. So our negative control for cytochrome C retention. So you can see here that everything is left shifted because there's no cytochrome C in the cell. So it's 100% cytochrome C release. And then because DMSO is our positive control for retention, we tend to normalize our samples after getting all the FACS analysis to the DMSO sample. This is an example of something I did last week, just an untreated profile of CDHL4, which is a double hit DLBCL non-Hodgkin's lymphoma cell line. So this is a typical readout you'll get for a BH3 profile. You can see here that we have the ALI as 100% cytochrome C release and DMSO is 0. So those are our controls. And then we look at the response of our peptides. So you see kind of a little bit of a dose-dependent response for BIM and BID, as well as PUMA. So this is a good response indicating that this cell, when exposed to activated forms of BID, BID, or PUMA, usually this cell line undergoes apoptosis, and we consider it rather primed. It doesn't have a large response necessarily to any one specific single inhibitor, but it does show some response to some peptides or drugs that inhibit multiple anti-apoptotic proteins. So then what we can use when you have your BH3 profile, when you're looking at it, is how do you determine what it means? From BH3 profiling, we essentially can determine 3 different types of classes of apoptotic block. So this is, again, simplified version of the intrinsic apoptotic pathway. So if we see that there is like this previous one here, a good response to initiators and sensitizers, then what we consider that as a class C block, which means on top here you see the class C. It means that the pro-apoptotic proteins are functioning fine, and the only reason the cell doesn't undergo apoptosis is because the anti-apoptotic proteins are acting on our effectors and activators, and preventing, or sensitizers, and preventing them from initiating apoptosis. Now there's 2 other types of block, which I'll show examples on the next slide here, which is class A and class B. Now these blocks are different because they're going to show a defect in the pro-apoptotic proteins. So no matter how much we get rid of or inhibit the anti-apoptotic proteins, these cells are still not going to undergo apoptosis because they have some sort of pro-apoptotic defect. So here's a reminder of the binding pattern on the bottom, and then these are experimental examples we've done in the lab showing different types of classes of block. So on the left, we have our class A block. So this class A block, what it's showing is that when we expose the cell to BIM, and to a lesser degree BID, we see cytochrome C release, meaning that the effectors backs and backs seem to be functioning fine as long as they can be activated by our initiators. But that means that endogenously, something is not working, something is wrong with BIM and BID, because we don't see any response to the release of these proteins with inhibiting the anti-apoptotic proteins. Now, class B is basically our most severe type of blocking C, and this indicates a problem with the effectors backs and back. Because basically, no matter what you give them, the cells that have class B blocks or samples that have class B blocks do not respond and release cytochrome C. So that means no matter what we do upstream in the pathway, the endpoint effector of pore formation by backs and back is not working. So it indicates some sort of defect in the effectors. And then here's a more, like we saw before, more typical class C profile where we see nice responses to both initiators and sensitizer proteins, as well as small molecule inhibitors like venetoclax. So this one has a very large 199 response, similar to the 737 response, which indicates that the majority of the anti-apoptotic contribution of this cell, which is a cell line called the LY1, is BCL2, is majorly responsible for this inhibition. So all that in mind, I know it was a lot quickly. How can you use BH3 for your research? Well, one, like I mentioned, you can identify the apoptotic priming of your cells of interest. So how close is this cell to undergoing apoptosis? Is it class B, which means it's very far away, or class C, where it's likely to undergo apoptosis when you give it some sort of drug or chemotherapy, for example. You can also correlate BH3 profiles to responses to treatment, either in your cells or in patient responses even, if you're using primary patient samples. So that's especially useful in our lab in chemotherapy, where I can analyze a patient sample with BH3 profiling and then look at how that patient responded to actual treatment. And you can kind of correlate that cells that are pro-survival and resistant to treatment, have these more or less primed. You can also experiment with different drug compounds and pre-treat cells before doing your BH3 profile with different, say, chemotherapy or inhibitor or drugs, and basically screen potential drug combinations that could synergize and increase apoptotic priming. And then something we've been getting into more recently in our lab is highlighting functional defects in the apoptotic pathway, most notably in pro-apoptotic proteins, which is in some disease, at least in non-Hodgkin lymphoma, kind of underappreciated in the field. So some examples of this type of work is shown here. So this is a project in my lab where we essentially profiled 124 non-Hodgkin's lymphoma samples. I won't go through all the details, but what is important is that here we're looking at the BIM and PUMA response, which is what we namely use because they're the most effective sensitizer and initiator to define a class A or B block. And in the samples red here is on both graphs are class B samples because they are not responding to either BIM or PUMA. And then we have class A samples that respond to, have in most cases, a moderate response to BIM, but then no response to PUMA. So this is an example of where we were able to identify, one, the level of apoptotic priming in primary lymphomas based on the average cytochrome C release response. So you can see that DLBCL has a much more variable response, and therefore we consider it less primed and more resistant to apoptotic insult stimuli than other non-Hodgkin lymphoma. But it also allowed us to identify these class A and B molecules, or these samples or patient population of that seem to exhibit pro-apoptotic defects, which is kind of something that remains to be explored in the field. And I put this in here, even though it's IF imaging, just to kind of highlight how The BH3 profile gives you an overview of functional capacity of apoptosis, but how it can be a jumping off point for your next experiment. So basically, when I saw that there was class A, B, and C molecules within DLBCL, or samples within DLBCL, the next thought is, okay, well, there's some sort of dysfunction going on. What is it due to a lack of protein, for example? So here I assess the expression of BIM and BAX in the same samples, essentially. And something interesting we saw is that the class A's had no expression of BIM and BAX. So the result of the result of the poor responses to BIM could be reflective of low endogenous levels of BIM and just a dysfunctional or reduced absence of these proteins. But in class B example, like in this example, we actually saw that the proteins were highly expressed. So that means there's some sort of other framework going on, and we're still exploring that. But it's just kind of showing you an example of how the BH3 profile kind of gives you an idea of maybe where you could focus your next efforts and other experiments. Here's an example of using BH3 profiling on cell lines to kind of look at synergizing of potential treatments that I had mentioned before. So R-CHOP is basically the standard chemotherapy regimen for patients with DLBCL. So here, we pre-treated cells with the different drugs listed on the x-axis, which are components of R-CHOP, in 2 DLBCL cell lines, and analyzed the change in response in venetoclax, our BCL2 inhibitor, and PUMA, our pan-sensitizer. So we call this change in response, shown by this example on the left here, delta priming. So basically, how much did we increase the priming of the cell? And what we were able to see here is that certain pre-treatment with these drugs before BH3 profile increased the responsiveness of these cell lines to venetoclax, specifically in mitotic inhibitors like vincristine, vinblastine, and MMAE. As well as changing the overall priming as seen by the PUMA response in the same number of drugs outside of doxorubicin and MMAE. So it's just another example of how you can use BH3 profiling to inform on the synergistic effects of different drugs in cell lines, as well as you could apply the same methodology to primary samples. And this is an example of correlating BH3 profiles from an older paper, not in our lab, it was an old Thai lab, to cell viability studies. So one of the important things about BH3 is that it can be used to predict how samples will respond to a drug. So just doing an untreated BH3 of these cell lines here, we see that CDHL4 and 6 have a class C block, and CDHL8 has an A block, CDHL10 has a class B block. So we would predict that exposing them to an anti-apoptotic inhibitor would mean that CDHL8 and 10 would not respond well because they're not responding to PUMA, which should inhibit the same protein. Whereas CDHL4 and 6 do respond, so we would kind of assume that, you know, inhibiting some of these anti-apoptotic proteins should lead to cell death. And you do see this correlation on the right. B is a negative control enantiomer, and A shows that as we would predict by the BH3 profile, 4 and 6 have reduced viability when exposed to increasing concentrations of this inhibitor compared to 8 and 10, which do not show any reduced viability and no response to the drug. So to kind of summarize BH3 and its adaptability is we use it as a flow cytometry technique, but it actually began as an ELISA technique, and it's being currently adapted into immunofluorescence as well as other high throughput screens and high throughput protocols for drug analysis. So basically taking the framework of interrogating these cells with different stages in the intrinsic apoptotic pathway, taking that framework and applying it to already known techniques and methodologies is basically what I'm trying to get you guys to appreciate here, because we can change the panel, change the kind of way we're reading cytochrome C release, all in this BH3 framework. So it's a very adaptable sort of mindset that allows you to look at apoptotic function. And on that note, the Latai lab recently published a paper this year discussing a new technique they call high throughput dynamic B cell profiling, or BH3 profiling, sorry, which is kind of summarized here, where they are taking fresh tumors, dissociating them, and then distributing them in large well plates. And then each well has a different drug or something they're incubating it. So it's essentially a very high throughput, like drug screening, to see how these cells, like which drugs will potentially respond in this sample, or you know, either cell line or patient sample, or not cell line, but you know, like a murine model or something of tumor. And what they did too to increase the sensitivity is, instead of doing a flow cytometry readout, they used an antibody here, and you can see the staining on the bottom, staining for cytochrome C in immunofluorescence, and then imaging at a single cell resolution the expression of cytochrome C, and then quantifying that. So you can kind of see at the bottom here how at the higher concentration of BIM that this cell doesn't stain positive for cytochrome C. That means it's releasing it. So it's just another way to kind of get the same data that we're seeing, but maybe at a more sensitive level than the flow cytometry. So to kind of summarize the BH3 profiling pros and cons, it's a functional assay that gives an overview of apoptotic function, especially the intrinsic apoptotic function. It's easy to do because it involves techniques that are pretty much already standard, like flow cytometry or immunofluorescence. It's adaptable. You design your panel specifically to your needs. You can do an abbreviated panel, you can do a larger one, and you can use different methods of analysis to kind of assess the function of your cells. So the cons are that it's an overview of the system, but it doesn't provide you necessarily with information at like a protein binding level or a specific interaction level. So it's kind of, in some ways, a jumping off point for future experiments. You do need sufficient cells if you're exposing to larger panels. So for example, in my panel, if I don't have like 5 million cells in a sample, like it gets harder to get a sufficient number of events to appropriately quantify my results. And also in primary samples, there's lots of variability in terms of just intrinsic cell death and responses. So you really need to do a lot of primary samples to get kind of reliable results. These are some resources, just papers referenced in the presentation that are useful to exploring BH3, as well as the link again to the Latai lab, which has all the information about starting BH3. So I'll open the floor to any questions you guys have. That took a little longer than I was expecting, but I apologize for that. But it's kind of it's a technique that kind of needs a lot of explaining sometimes.
Thank you for the presentation. I really enjoyed it. I guess I could start with a question if no one else has them right now. Maybe it's a silly question too, and it might be like too specific. But the digitonin, how does it permeabilize membranes again? How does it act?
So digitonin forms, I mean, I'm gonna blank on the actual chemistry of it, but it forms basically like, I think, ion channel membranes on the cell surface, but they're not. It's not completely destructive necessarily. So you're going to preserve your cell shape, but without actually destroying the cell.
And it'll. Oh, sorry.
Yeah, go ahead.
And it'll permeabilize all the cells homogeneously? Because do some detergents kind of rely on interactions with cholesterol? And if cellular membranes have different levels of cholesterol, could they be permeabilized in slightly different ways? This might be like way too specific. Sometimes I focus on like nitty-gritty details that don't actually matter.
No, that's fine. So yeah, I haven't had any issues with digitonin in terms of permeabilizing my cells. But if you have a specific cell that potentially has those like contraindications or reactions, then there are potential other detergents you may want to look at. And it's mostly about just adapting the protocol to fit your needs, right? But in most cases, I find digitonin works fine, and used at the appropriate concentration, every cell is appropriately exposed to the peptides.
Thank you.
And that's why you have like sort of your controls and stuff to confirm that. Because if I'm not seeing a response in my controls, because every well was given the same cell, then that's one of the potential areas that it could have gone wrong, is that the cells weren't permeabilized appropriately.
I have another question. I don't know.
Sorry. If nobody else has a question, you can go ahead.
Just with that last slide, that publication from the Latai lab and their high throughput like image analysis. So I guess the way they analyzed the images is they counted the cells with the DAPI channel and then just quantified the green fluorescence intensities.
Yeah.
Because I know it looks obvious in the field of view that you show here, but I guess like the cellular coverage is different in each image. So and with image analysis, sometimes you take it for granted. Like it can be a little bit tricky sometimes. So I'm just curious do you know how they did it?
Yeah, I'd have to look over the paper again. I was just kind of introducing this as because it's a relatively new paper, just kind of introducing it as a way that you could apply the same framework to a new type of protocol. So I thought it was interesting that they were able to kind of do an immunofluorescent version of BH3. But yeah, they would need to have some sort of normalization or control, as you said, because just, variability and like say if they're measuring the MFI, it could make you believe that cytochrome C is being retained more when it's not really that. It's just, some minor differences in fluorescence.
Right. And I know too, sometimes I had issues with background, at least when I was doing immunocytochemistry, like the illumination, like the background can change from the center of the field of view towards the edges. So I might look at their protocol to see what corrections they did and if they used any programs to automate that, because it would take a lot to analyze all of those wells, like 384 well plates. So it'd be nice to know how they did it.
Yeah. I don't have the whole protocol here with me, but yeah, they claim to basically be able to kind of analyze thousands of drugs in like a relatively short amount of time, and so they definitely have to have some sort of especially at the imaging stage, because and quantification, some sort of efficient way or normal or standardized way of doing it, because I mean, it can take me weeks to do image analysis on IF.
Right? Yeah, same. Cool.
Okay, any other questions, or should we wrap up here?
I think I'm good. Thank you so much for the presentation. It was very easy to follow and very interesting, and I'm glad you went into the depth that you did. So thank you.
Yeah, it went a little longer than I had intended, so I hope that didn't bother you guys, and I hope it was easy to understand, because I find sometimes trying to go through BH3 with the methodology and like understanding what exactly we're looking at, I find it can get kind of lost and confusing sometimes. And hopefully I didn't lose you guys too much. But if I did, feel free to reach out to me if you want to kind of discuss BH3 more in depth or discuss a plan to potentially do an experiment. I'm more than happy to help.
Yeah, I think that was well done. From the non-immuno, I mean, I'm in HIV, so it's very different from my current field, but I could see the applications, and I think it was well explained. I mean, an application outside of apoptosis, like they are relatively easy to think about from that perspective if we have a similar readout to cytochrome C, but I'm sure those could be found in individual fields. So yeah, well done.
Absolutely. Okay, well, if there's no more questions, then I guess we'll wrap up and just like thank everybody for attending, and we should have a recording of this up on EMGSS's website as well. If you want to refer to this talk, you can see it there. Otherwise, just send me an email if you guys want to discuss any BH3.